Well, hey AP Biology, hope you're doing well when you watch this video, and I hope you're having a good day. This is part one of Mendelian genetics, our genetics unit. Of course, due to the storms in Florida and all of that, we need to do these notes online to try to catch up and do our best. So I'm going to do my best for you. Of course, we'll practice in class and go over questions and examples and all of that. And so thanks for joining me today. I know, of course, this is an assignment, so not too much of a choice on your part, but there's always a choice, so I appreciate it. So inheritance, genes, and chromosomes. So right now you see two Jewish priests, and on the right are the ordinance of circumcision being carried out. And there's a story from the past, and it was a case study in one of my older books, and basically a woman gave birth to a son. She brought him on I forgot what day, the 7th or 8th day, to get circumcised, and he couldn't stop bleeding after they circumcised him. And so tragically he perished. Fast forward, she has another son, and, of course, following the law, brings him to be circumcised. Bleeds out on the table again, tragically. Loses her second son. And so fast forward, and she gives birth to another son. And the priest showed wisdom and was like, listen, he's not going to be rejected from the covenantal family and the faith, but we are not circumcising him because something else is going on. Something inside is wrong. Something with the genetics or DNA or something. And so what they saw was they had insight into that. And so what they had insight into was actually a genetic disorder that we're going to talk about today. And so this is the learning objectives. So these are the learning objectives, like I just said, about where we're going here. Okay? And so heritable information provides a continuity of life, like what you receive from your parents, right? And then lines of descent from common ancestors. We can trace our DNA all the way back to the beginning. And so explain how shared, conserved, and fundamental processes and features that support the concept of common ancestry for all organisms. That's evolution and the importance of that. But you can see right here that DNA, of course, ribosomes are found in all forms of life. We can bring up the endosymbiotic theory that we talked about. Major features of the genetic code are shared. Anytime you see that word like shared, and shared and all that is a symbol and a sign that we're talking about the continuity of life. Core processes are conserved. That's another key word, like conserved. The next learning objective is explain the inheritance of genes and traits as described by Mendel. And then you can see that here's some of the stuff that we need to talk about when it comes to Mendel's laws of segregation and independent assortment. Then fertilization, of course, the fusion of two haploid gametes. Remember meiosis, right? Creating 4 unique, genetically different haploid cells, sex cells. Those cells, sex cells, sperm and egg, combine to form a zygote. And then, of course, probability rules. We'll briefly talk about that, but that's basically, understanding that if they're mutually exclusive, you can add them. But if they're independent, then you have to multiply them, and we'll talk about that in a second. That practice is really brought up from the Punnett squares and understanding genetics and all that. You will need to know monohybrid and dihybrid crosses, sex-linked, which is Thomas Hunt Morgan, possible stuff as well, and genetically linked genes, which is also Thomas Hunt Morgan. And that can be often predicted from data, including pedigrees, which we'll dive into, not this lesson, but the next one. And so this leads us to, a little bit of a biblical worldview. I of course, being a follower of Christ and teaching at Christian schools, it is a joy to be able to talk about this. And so circumcision. Paul has something very interesting to say about circumcision, and I want to show it to you. Let's go right here if it brings up. Okay, good, nice. And so here is circumcision in Romans 2:25-29. And notice what he says. For indeed, circumcision is of value if you practice the law. But if you are a transgressor, a breaker of the law, your circumcision has become uncircumcision. He's like, listen, Paul's like, listen, it doesn't matter what you've done if you're not following the law of God. So if the uncircumcised man keeps the requirements of the law, will not his uncircumcision be regarded as circumcision? And he who is physically uncircumcised, if he keeps the law, will he not judge you who, though having the letter of the law and circumcision, are a transgressor of the law? For he is not a Jew who is one outwardly, nor is circumcision that which is outward in the flesh, but he is a Jew who is one inwardly. And circumcision is that of the heart, by the Spirit, not by letter. And his praise is not from men, but from God. God wants his children, God wants his followers to be Jews inwardly and have circumcised, completely changed hearts by his Spirit. And that's what regeneration is. Surrendering to God, surrendering to Christ, and then letting Christ completely give us a new heart. Even in the Old Testament, there's multiple passages about this where God is focused on the heart. He's not focused on outward obedience as much as inward surrender and inward faith. And so, of course, inward faith will produce outward obedience. But if you have the obedience on the outside, Paul's like, listen, if you have the obedience on the outside, but you're transgressing the law, that doesn't matter. You're showing what kind of heart you have when you're breaking God's ways and breaking God's law. And so that is such good news for us, that God wants us to be changed from the outside, from the inside out. And so this is, let's go back to this. And so this is how hemophilia is inherited and why is it. So this is Queen Victoria's disease. And so that's what that child suffered from, hemophilia, which is a bleeding disorder. And it's carried on the X chromosome. And we'll talk about Queen Victoria down the road. So if you want to pause right now and think about it, genetic testing and genetic disorders, and in this crazy kind of sad world we live in, there's a lot of... can I change the position of this thing? There's a lot of possible concerns when it comes to these things. So think about some of the potential concerns or issues. Like, what if your employee knew your genetic code? What if your employer knew your disorders, possibly? Or maybe you're more prone to cancer. What if your insurance company knew that? And so just kind of interesting stuff. So here's the big idea. The meticulous work of Mendel and others provide evidence for a particulate mechanism, like there's actually particles that we inherit from our parents. And this led to our current understanding of a chromosomal basis of inheritance of genes from parent to offspring. And this provides structural and functional evidence to support the relatedness of all domains. Of course, evolution by natural selection teaches that life came from one single cell, all domains of life. And so, get ready for this chapter. Describe the chromosomal makeup of the diploid cell. Describe how a gene codes for a protein. That's just in general. You should think about like DNA leads to RNA, leads to proteins. What are those two terms between them, right? What's the term from DNA to RNA? And what's the term from RNA to protein? We haven't dove into the mechanics of that. That's more Unit 6. But nonetheless, explain how a protein structure determines its function. Oh man, we've been talking about it all year. Shape leads to function. And so sickle cell is a disease that we'll kind of bring up in genetics and stuff. So here's an infamous probability problem. Testing your probability intuition, the two-child puzzle. Okay. Of course, we're not working in Paris Code doing notes at home, but we talk about, practice problems in class. But here we go. A family has 2 children, a newborn and a toddler. At least one of the children is a boy. What is the probability that the other child is a girl? So I want you to pause this problem, think about it. So pause. And so you can see right here that the answer is 2/3. Most people want to say 1/2. But if you write out the possible outcomes, and since you know that one child is a male, you can cross this one out. Okay? And so now, it's 2/3. And so the key to understanding this puzzle, and obviously, crosses, is that there's multiple ways to produce a boy and girl combination. Girl first or boy first. There's multiple different ways to achieve a combination when it comes to genetics. So in the mid-19th century, we're trying to figure out, how does genetics, how do offsprings work? And so there's a blending inheritance theory. Gametes kind of contain determinants that blended, like a tall and short person give like a medium-sized person. But then there's also particulate inheritance, is like physically distinct and remained intact. And so good old Mendel did studies with his beautiful garden peas, and that clearly supported the particulate theory. Why did he use garden peas? They were easy to grow, easy to manipulate, quick life cycles, and so very easily to control, reproduction as well. And so easy to cultivate, short generation time, self-pollinating but can be crossed, and then true breeding varieties were available. Simple, objective traits. Good choice. And so here are the flowers, came purple or white, beautiful flowers. And so where's Mendel? Oh man, here's his band of bros, and there's Mendel looking at a pea plant, and this guy's kind of like, man, I don't know about him. This guy's also kind of looking at him kind of strange. I'm not really sure what that is about. I don't know what this guy's looking at. This guy's deep in thought. Anyways, but there's Mendel, and so he had some time, of course, to carry these out. And so peas normally self-fertilize. So it is helpful to understand basic, plant anatomy here. Stamen, that's the male part of the flower, men. The pistil and ovule are the female parts. There's the eggs inside. Pollen is the male sex cell, so you can see right there. Pollen will land on the pistil, then through a complicated process kind of burrow in and deliver the sex cells, and then fertilization will occur. Okay? And so stamen, male part, carpel is the female part. Sticky stigma, that's where the pollen lands. Style, and then ovules in the ovary. And so characters, physical features, flower color, seed shape, traits. We'll kind of do an activity of this in class with human characteristics. Traits are forms of a character. So a character would be like flower color. And then in pea plants, the forms would be purple and white. Character could be seed shape. And the traits would be like round or wrinkled. Okay, and so Mendel first worked with true breeding varieties. Okay, so true breeding varieties is important to think about, and so that's what he first dealt with. So no matter how many crosses, they always gave the same outcome. Okay, this is a website I encourage you to go to. I'll show you it in class. It's a beautiful website when it comes to genetics and DNA and everything like that. I've used it for many years. And so here's what Mendel did, right? So he removed the stamens. He controlled which flower was going to pollinate. And then he transferred sperm-bearing pollen from stamens of white flowers. So this is a true breeding white flower. No matter what, it produces white. And no matter what, this produces purple. And he pollinated the purple. Waited for pollination, carpels to mature into pods, planted the seeds, and then what happened? Well, in the first folial generation, F1 generation, you need to know like that term, the result was all purple flowers. There wasn't like a lighter purple. There wasn't like a blending. It was all purple. And so Mendel was like, well, this is interesting. So this is the F1 generation. And so he just used those true breeding plants. And he occurred in two distinct alternative forms. So like the garden pea, I mean, we would say that the Lord really blessed Mendel in wisdom in choosing this. Mendel, of course, is one of the greatest scientists ever to live. I'm pretty sure his work was not discovered while he was alive. His work was discovered after he passed away, and so he never really got credit for the importance of his work on this planet. But of course, his legacy lives on. And so the parental generation and the F1 and F2s are, filial means son, and so the hybrid refers to offspring of crosses between organisms differing in one or more characters. So hybrid, true breeding, you need to kind of understand these terms. And so in the first experiment, Mendel crossed plants differing just one character. So that's called a monohybrid cross. And so this is a similar type of thing when he was like, okay, I'm going to take true breeding round seed. No matter what, this plant gives round seeds. No matter what, this plant gives wrinkled seeds. Two different types of shapes here. So these are traits. Pollinated them. Okay, of course, controlled which one, grew them, and then all he saw were round seeds, no wrinkles. Okay, and so that's the F1 generation. And so Mendel then was like, all right, well, I'm going to take the F1 generation and pollinate them. So I'm going to pollinate this plant with itself. And then what he saw was that the wrinkle came back. Okay, and so the hypothesis is there's no irreversible blending, right? The wrinkled came back, and so with Mendel, he thought, okay, well, where's the wrinkle? The wrinkle is hiding somewhere here. Like the gene, the particle. I don't know if you thought of genes. Of course, genes they would discover later, but some sort of particle that codes for wrinkled is hiding in those smooth pea plants. Okay, and so the hypothesis was rejected. So Mendel's findings led to rejection of that. The trait did not disappear. It came back. Okay, so all 7 crosses between varieties with contrasting traits gave the same kind of data. Mendel was meticulous in data collection. It's amazing the data that he collected. I'm going to show it to you here in a second. And so this led to some terms, of course, dominant and recessive. Dominant overpowers the recessive one, and dominant shows up in the phenotype and genotype. And the ratio of dominant to recessive was 3:1. Just real quick, I'm sure I'll talk about it later. But phenotype, let me see if I can type with an old keyboard, is what you physically see. And genotype is the genetic makeup. Okay? And so what he physically saw was the ratio come back. And so he saw this 3:1 ratio. So here we go, purple and white, reviewing what we just talked about. And then the F1 generation was all purple plants. So he crossed the F1s together, and he saw the white come back. And look at that data, right? Look at that data that he collected right here. 705 to 24, that's roughly a 3:1 ratio. And of course, we just did our chi-square test, so we could always do a chi-square analysis based upon, hey, are the choices, does this fall within the realm of, accepting or failing to reject, the null hypothesis? And so you can see how hopefully the chi-square can be applied easily to genetic problems. Mendel proposed that genetic determinants occur in pairs and are segregated, which means they are separated. So each plant has 2 genes for each character, 1 from each parent. So we now use the terms, of course, diploid and having 2 copies, and haploid. Remember, meiosis creates haploid sex cells. Mitosis is diploid clone cells. And so different traits arise because there can be different forms of a gene. Different forms of genes are called, you need to know it, are called alleles. Alleles are different forms of genes. And so we, as human beings, have lots of different alleles. There's the phenotype and genotype definitions, which we already just talked about. And so when we write this stuff, and I know y'all have done this earlier on in 9th grade and middle school and maybe even before then, but this is heterozygous, difference, hybrids, and these would be considered true breeding, okay? That would be round, right? And then wrinkled would be little r, okay, because that would be the recessive trait. And so, here we go. This is purple. So what you saw was a 3:1 ratio, and we now know the genotypes is 1 to 2:1 ratio for genotypic ratios because there's 1 homozygous, 2 heterozygous, and 1 homozygous recessive. And so 1 to 2:1, 3:1 here. And so homozygous means the same. They carry 2 copies of the same allele. Heterozygous means they carry different alleles. So here's round and wrinkled. All of them were round, but all of them were heterozygotes. And then when you cross that plant together, you can either get this allele or this allele in a sex cell due to meiosis, and then you have a 3:1 round to wrinkled ratio, but notice that the genotypes are 1 to 2:1 ratio. Homozygous dominant, heterozygous, homozygous recessive. And so this is his data. I mean, look at this, right? I mean, we get frustrated when a TikTok video doesn't load correctly. That meticulous data collection, right? And you can see the ratios. They all are so close to 3:1. And so he realized that there was some sort of law of nature governing this. And so purple times white, yellow is the dominant seed color, round's the dominant shape, green's the dominant pod, inflated, axial, which means on the side, not at the end, and then tall versus dwarf. It's amazing. So 3:1 ratio. And so this is just showing you kind of alleles. So there's one location for a gene, and that gene can have multiple different alleles. And so this DNA sequence results in the absence of an enzyme. And so that absence of the enzyme prevents the purple color from being, pigment from being synthesized correctly, so therefore it's white, right? Of course, so this is a huge concept where you need to understand everything that we physically see in an organism, as far as a trait goes, is genetically determined. So we'll talk about traits in class as far as human beings go and stuff, but it's amazing to think about. So here's Mendel's first law. The law of segregation states that the two copies of a gene separate, and that's the key word, separate, when an individual makes gametes, and that's due to meiosis. So we've been talking about Punnett squares, and here's just an example of a monohybrid cross, which you could fill this out, on your own. But of course, this, anything right here goes in these boxes. Anything right here goes in these boxes. And so that is how you fill it out. I'll show you an example here in a second. And so there's the example, right? So when you're crossing the F1s, you could either give the dominant or the recessive in the sex cell, since it's the same plane, dominant or recessive, and then you just fill in the boxes. Little t goes here, capital T. Always write the capital first, little t, and then little t, that would be tall versus dwarf. And so Punnett squares predicts on paper 3-to-1 ratio, and then we would test it out in real life, chi-square test, see if it matches up. Genes are now known to be relatively short segments of DNA, found in much longer DNA molecules that make up chromosomes. Different alleles can segregate versus that. Okay, so genes determine phenotypes mostly by producing proteins. Remember, shape determines function. By producing proteins with particular functions. So shape determines function. So here's just a great example. Like the wrinkle P phenotype is due to a lack of starch branching enzyme. So why is it wrinkled? Because it doesn't make a starch like as the way the round seed does. And why is that? Because it's lacking an enzyme. It's lacking a functional enzyme. And so hopefully you can understand that phenotypes are due to genetic differences in these pea plants. So Mendel verified his hypothesis by doing test cross. A test cross is always done with a homozygous recessive. Okay, that's what it's always done with. So if I ever bring up test cross, if we're dealing with plants and stuff, you always take a organism that is homozygous recessive and you cross it, because basically with the test cross, you're wondering, is the parent plant this, like all around, or is the parent plant that we're wondering about heterozygous? And so on paper, if you cross this, hopefully you can visualize that if I cross this, if I cross oh man, that was not good. If I cross two capital R's and two little r's, well, every box will get a capital R, so it'll be round. But if I cross a heterozygous capital R and little r with two little r's, then 50% on paper. Okay, and so here's what it looks like, right? All will be round here, 50% here. And so then you can draw your conclusions. So you kind of work backwards, trying to figure out the parents. Okay, so here's just another picture here, like, oh, is this like homozygous dominant or heterozygous? So I don't know. Let's cross it with a test cross. Homozygous recessive, 50%, 100%, and that's how you figure it out. Mendel identified the second law by following two characters at the same time. Crossing two true breeding parents differing in two characters produces dihybrids in the F1 generation. Di means two. So dihybrid cross can determine whether two characters are transmitted together or independently. And so oh man, what happened there? I didn't know we could do that. Sorry. And so here's the true breeding parent. So here's two characters, right? So this is the dihybrid cross. We'll practice these in class, but let's just kind of get our feet wet right now. So these are two homozygous, right? Homozygous. So this round and yellow, and this is wrinkled and green. Okay? And what he saw was this, all round yellow plants. So then he crossed, he self-pollinated the F1 generations. And Mendel asked whether, okay, in the gametes produced, would the traits be like go together or would they segregate independently? And of course, through divine providence, we would say like he chose an organism that they segregated independently. There are some organisms. Thomas Hunt Morgan talks about this, and he sees it with various things. And Thomas Hunt Morgan also deals with sex-linked traits, but he saw like linkage in a different model organism, which I won't get into right now. And so, two possible outcomes. If if the alleles were linked, gametes would be this or this because they would travel together. So it'd be a 3-to-1 ratio. But if independent, you could get 4 possible gametes. You could have capital, 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 lowercase, 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 or lowercase capital. And so this famous ratios, you need to know these ratios, by the way. Like this is famous ratios with Mendel. 3-to-1 and then 9-to-3 to 3-to-1. Famous ratios that you just need to know. And so what he saw was this. He saw a 9-to-3 to 3-to-1 ratio. Out of 16 possibilities on paper, he saw this in real life. And so he realized that these gametes independently assort. They're not linked together. And so this, how you figure out this is FOIL. I'm not writing with a writing pad right now, but I'm going to do my best to try to do this with simple letters. So this FOIL is this. If I had a capital T and lowercase t and like I don't know, like a capital R and lowercase r, what I mean is that how do you figure out possible gametes? Well, you figure out possible gametes, I'll do it in different colors, by taking the first letter between each character, each gene, right? So this is, so capital T, lowercase t is the genetic combination for one trait, right? Tall, height. So you take the first one, and then you take the outside letters. So you could get capital T, capital R, or capital T, little r. And then you take the inside letters, little t, capital R, or the recessive T could also go with the recessive R. And so that's how you get 4 possible gametes. First, outside, inside, last. Okay? And so never, do extra work. So if your gametes are ever the same, don't write that on the Punnett square. That's wasting time. And so alleles of different genes assort independently. This law does not always apply to genes near each other, which is what Thomas Hunt Morgan found. And so how does this work out in meiosis? Well, you have, prophase 1, which is where crossing over happens. And you have these possible combinations, based upon, independent assortment. So here's your concept check for our first section here, and I'm briefly going to talk to you about probability. But pea plants heterozygous for flower position and stem length are allowed to self-pollinate. So you're crossing that genotype with itself. 400. Draw a Punnett square. We'll definitely go through this. And how many offspring would be predicted to have terminal flowers and be dwarf out of 400. So you're going to use the ratio, the probability, and figure out of 400 on paper how much would you expect to find. List all gametes. So think about FOIL here, although there's 3 different types, right? So that's just kind of trying to think a little bit outside the box. Of course, think about our complexity and how we have 20,000, or I believe something like 20,000 different protein encoding genes. I could be a little high on that, but it's just think about all the different combinations, right? Such complexity, such beauty. In some pea plant crosses, the plants are self-pollinated. Is self-pollination considered asexual or sexual reproduction? Yeah, I want you to definitely think about that. We will definitely talk about that one because that was a big misconception last year. Okay, and then probability. Just briefly diving into this because probability really happens when you practice it. But what's the probability of flipping a coin on heads? Hopefully you said 1 out of 2, 50%, right? But what's the probability of flipping it in heads twice in a row? Well, then you would take 1/2 times 1/2, and it's 1 out of 4, right? And this is where you get like the insane probabilities of winning the lottery. What's the probability of picking a heart out of a deck of cards? Well, 52 cards in a deck, 13 per suit, I believe, so that's 1 out of 4, right? But let me tell you a brief story about probability. This is roulette, and so it's definitely it's an interesting game. I was trying to go into like how it works, but it doesn't really matter. But obviously you have red and black, right? But the phrase "the house always wins." Well, there's a reason for that because there's green on the board, right? Green shouldn't be there. It should be red or black. 50% if you land on red, or 50% if you land on black. But nope, that's not good enough odds for the house. The house is all about numbers. It's all about what's on paper. So the house is like, I'm going to add some green to make sure I win more often. And so I won't take up your time telling the story about the one time I played roulette and actually won some decent amount of money. But I went back the next day and lost because why? The house always wins. But yeah, the biggest payout you can get in roulette is, of course, probability is the likelihood of an event occurred. The biggest payout you can get on roulette is landing on a single number. It's like a 1 to 35 payout, right? So if you bet $1, you get $35 in return. But look at that percentage, right? 2.63%. But look at the probability here. And there's famous bets in history if you want to look it up, like, a guy sells everything and puts all of he got on red and he wins or loses, and obviously tragic things happen sometimes if they lose. But 46%. 46%. Why? Because the green's there. Why? Because the house always wins. And so Mendel's rules reflect the rules of probability. The outcome of one coin toss has no impact on the outcome of the next one. In the same way, the alleles of one gene segregate independently. And so there's a couple of different rules. Multiplication rule states that the probability that two or more independent events will occur together is the product of their individual probabilities. So F1, sorry, this is kind of overlap, but each gamete has a half a chance, 1 out of 2 chance of carrying the dominant allele, and it has half a chance of carrying this. And so you have half chance, 1 out of 4. So of course, you're like, well, we just did this on paper. But yeah, this is just understanding the math behind it. The addition rule states that probability that any one or two or mutually exclusive events will occur is calculated by adding them together. And so this can be figured out that an F2 plant from a monohybrid cross will be heterozygous rather than homozygous. We can apply the rules to predict outcomes, calculating the chances, and various things like that. And so, for example, if you cross capital Y's, 1 out of 16, 1 out of 8 if you do this cross. And this is just all, of course, trying to figure out the math behind the Punnett squares, okay? So for these, you could try your best on them. We'll have some, examples in class as far as probability goes. But like I said, probability is mostly just practicing genetics on paper and understanding like how it all works, okay? So that was part 1 of genetics. Thanks for hanging in there with me. Hopefully it helps. We'll practice in class. God bless.